Hello, everyone, and welcome to the July entry of our ALS Canada Webinar Series. Thank you for being here. I hope everyone's having a wonderful afternoon. My name is Dr. David Taylor, the Vice President of Research for ALS Canada, and I'm very pleased to present our speaker for today, who is doing something as a first for us. So we're going to have a webinar now in English, and our speaker is kindly going to do a webinar as well en français afterwards as a separate webinar. So this will be the first in our ability to be able to provide in both official languages. So we're really grateful to Dr. Vincent Picher-Martel for doing that. Now our speaker today is going to speak about models in the laboratory and how we can help to convert those and understand from those into ALS therapies. Our speaker is Dr. Vincent Picher-Martel. He is going through the training as a medical doctor currently, and is currently also pursuing a PhD at Université Laval in neurosciences with Dr. Jean-Pierre Julien. Can you hear me, Vincent?
Yes, I can.
Perfect. Welcome and thank you for being here. The floor is yours.
Okay, thank you, David, for the presentation, and thank you to all of you who will listen to my webinar. So today I will speak about my PhD project about the pathological role of Ubiquilin-2 and its relevance for modeling the human disease in mice. So like David said, I'm a PhD candidate in neurobiology with Jean-Pierre Julien, and also a neurology resident in my second year of specialty. So for those of you who do not know, ALS, also called Lou Gehrig disease, it's a progressive loss of upper and lower motor neuron, upper and lower motor neurons, which lead to paralysis and eventually death within 2 to 5 years. There's two forms for the disease, the sporadic and the familial form. For the moment, there's no efficient treatment for the disease. We can give to our patient the riluzole, which increased the lifespan from about 1 to 2 months. And recently, the edaravone was accepted by the FDA in the USA for the treatment, but it's not yet available in Canada. There's also about 30% of the case of ALS which are associated with frontotemporal dementia. In that dementia, we can see behavioral abnormalities or personality change. So there's a lot of genes that we know to be mutated in the familial form of the disease. The first one was discovered in 1993 was the SOD1 gene. Since this time, many other genes have been discovered. In 2008, TDP-43 gene certainly changed research in ALS. And more recently, the C9orf72 gene have been shown to be mutated in 30 to 40% of the familial cases. So it's quite an important gene. And also in 2011, the Ubiquilin-2 gene was discovered. Ubiquilin-2 mutations are not so frequent, but the role of the protein is quite important in all the case of ALS. So I will talk about that later in the presentation. We know many mechanisms which are implicated in the disease. However, we are not able to link all of these mechanisms yet, and we are not able to explain why they are happening in motor neuron. But by example, we know that there is some problem in the clearance of the cells. There are mainly two mechanisms who are implicated for the clearance of the cell, the proteasome and the autophagosome. These two pathways are deficient in ALS because of different genes. We also know that the skeletal of the cells can be changed. Here we see at the right. We also know that cells surrounding the motor neuron are implicated in the disease. We think that both microglial cells and astrocytes can be toxic for the motor neurons. And one of the most important ones remain the aggregate formation. Aggregate are small stack of proteins who are in the cytosolic of the cells, and we think that these aggregates may be toxic for the motor neuron. One of the major components of these aggregates is TDP-43. So why do we need animal models for human research? So first, for sure, we have to understand the mechanism of the disease. So for that, we can use either cells or animals. But for sure, the animals may recapitulate more the complexity of their organs and the metabolism of all the cells. So to test drug, we have to test it first in animal, and we have to test it for the efficacy, the security. We have to check for the doses, the metabolism of the drug to be sure that it does not accumulate in the animal. And to create animal models, I will discuss about the way to do that, but we can use genetic mutation, exposition to toxin or other proteins. And we can use a lot of different animals. So the first most used one is certainly the mice. We use the mice because it's small, it's not really expensive to take care of. We don't need a lot of installation and big laboratory to use them. They are quite resembling to human. The organism of it's quite look-alike, and they have a complex nervous system. So it's a great model to mimic the disease. We can also use rats. They are almost the same thing as the mice, but they are a bit bigger. So we are able to perform some techniques who are difficult in mice. By example, intrathecal injection can be tough in mice, and it's more easy in rats. We can use monkeys. Monkeys are certainly the most resembling to human. However, they are really expensive to take care of. They need big laboratories, so it's really tough in Canada to do monkeys research. And also, they carry some disease that can be transmitted to humans, so it can be dangerous sometimes to work with monkeys. We can use more genetic model as the Drosophila or zebrafish or the nematodes. These models are mostly used for genetic analysis. They are also useful to screen drugs fastly. We can try a lot of drugs in one day and see which one could work the best. Also, the nematodes here at the middle are nice because they are transparent. So we can tag a protein with a color, a specific color, and we can track the protein in all the body easily. We can also use one cell like yeast, or also frog, pigs, dogs, or cats. They are less used, but it's interesting because the dogs are the only mammals who naturally develop ALS. Some dogs carry the SOD1 mutation and develop ALS. So how do we generate an animal models? So I will try to explain it simply. So the most of animals that we use are transgenic animals. So transgenic mean introduction of a foreign DNA. So for that, we use a plasmid, who are round sequence of DNA that we found in bacteria. So we take these plasmids, and we are able to cut at a specific point with enzymes and introduce the gene of interest. For example, if we take the Ubiquilin-2 gene, we are able to introduce it in the plasmid easily. After we got the plasmid, we take pregnant mice, and we take the fertilized eggs from the mice, and we just inject the plasmid into the fertilized egg and reintroduce the eggs into the mother. So the mother can have different children, and by genetic analysis, we are able to spot which one of them has the genetic mutation, and we are able to create a line with the mutation by crossing the mouse with other's mouse and to have a line of transgenic mice. We can also do almost the same technique for knockout. Knockout, in fact, is to eliminate the expression of a gene. So instead of introducing a new gene with transgenic animals, we are knocking down, so we are eliminating the expression of the protein. Another way is to use virus. So we can introduce plasmids into virus and infect the mice with this virus, so the gene can be expressed in all the cells. Another way is to the use of toxin. One of the best examples is the MPTP for Parkinson's disease. It's really an interesting story because MPTP was discovered in 1976 by a chemistry student in Maryland. In fact, this student tried to make illegal drugs, but instead he made MPTP, and he gave Parkinson's to a lot of persons in America. So since this time, we are using this molecule to mimic the disease in animals. So what is the relevance of the current animal models that we have to mimic ALS? All the data that I will present are from my review that I wrote with Jean-Pierre and Nicolas in 2016. So the first animal models that we mostly use are the SOD1 animal models. There's a lot of different mutation in the gene that we can use, but the most used one is the G93A SOD1 mice. This mouse has a lot of features resembling the ALS disease. They are losing motor neuron, they got paralyzed, they die of the disease, they got aggregates with neuromuscular junction loss. Neuromuscular junction, here it's the link between the neuron and the muscle. So they have a lot of interesting features. However, they are less accurate for modeling all of ALS case, because the SOD1 pathology is not found in every cases of ALS. This is a highly controversial subject, but most of the cases do not have SOD1 pathology, but have TDP-43 pathology. And in the animal models, there's no TDP-43 pathology. So the SOD1 models can mimic maybe 5% of the human case. In fact, we tried a lot of drugs in SOD1 models, and most of them, if not all of them, failed in clinical trials. So it's raising a lot of questions about the efficacy of this model to mimic the disease. So in front of that, we turn to TDP-43 models. TDP models have a lot of advantages, but also they are bad at some things. So most of the models develop an early age onset, so they die before 4 to 8 weeks of age. So it's not what we found in human. Most of them do not have, in fact, motor neuron loss. They do not have big TDP pathology, or they do not really die of paralysis. In fact, in posteriori, we are able to see that the mice are dying of gastrointestinal disorders. So it's not exactly because of the disease. So in our lab, we have generated a few years ago 3 different mouse lines with mutation in TDP-43. Our mouse line are developing an age-related disease, but they do not get paralyzed. However, they have a strong TDP pathology, they have decreased axonal caliber, they have motor dysfunction and cognitive deficit. So they are great models, but they are not perfect models either. More recently, C9orf72 animal models was published. One of the great one is published, I think, in 2015, end of 2015, show neuromuscular junction loss, reduced axonal size caliber, motor neuron loss. So these are all features of the disease, great features of the disease, but they show pathology which resembling C9orf cases. It's not yet clear if we can see RNA foci or DPR in other cases of the disease. So they are quite specific to C9orf mutation also, as the SOD1 are. There's also some FOS animal models. These animal models have the characteristic to die early, so they are dying before 3 months of age, so it's not an age-related disease, and they do not have really a motor neuron loss, or they do not have FOS inclusions. So these models are not a perfect model either. So there's also Ubiquilin-2 model. So one of the good, Ubiquilin-2 model was published in 2016 by Naut T. et al. So this model has great features also of the disease. It became paralyzed. It's dying of the disease. It's having muscular atrophy and also TDP-43 pathology. One of the bad points of this mouse is that there is a high variability between the mice. Some like 10 to 20% of the mice only get paralyzed and die of the disease. So it's not a perfect model, but I really like this model either. So what is Ubiquilin-2 exactly? Ubiquilin-2 is a ubiquitin-like protein which is implicated in the degradation of protein. So when you have too much protein or too much toxic protein in a cell, the mechanism has to be used to clear it. So one of it's the proteasome pathway, and Ubiquilin-2 make the link between the protein who has to be degraded and the proteasome pathway. So it's like a shuttling protein. It has been discovered in 2011 to be mutated in certain familial cases. Either the mutation is rare, it's like 1% of the cases. The presence of Ubiquilin-2 in the aggregates is prominent, and it's present in all the cases, even the sporadic ALS and other form of familial cases. So we found co-aggregation of Ubiquilin-2 with TDP-43 in almost all the cases. So it shows its important role in the disease. So Jean-Pierre and I have shown in 2015 that when you overexpress Ubiquilin-2 into neurons, you are able to induce a mislocalization of TDP to the cytosol. In fact, TDP is normally located in the nucleus, like I explained before, but it's creating aggregates in the pathology. So we show that when we overexpress Ubiquilin-2, we induce a strong mislocalization of TDP-43. And we think that these aggregates are toxic for the neurons. So in front of these results, we decided to generate a new model, to ameliorate our model, to have a better model to test therapeutic approaches and to study the mechanism underlying the pathology. So like I explained before, we generated a plasmid with Ubiquilin-2 mutations, and we inject it into mouse and generate a new Ubiquilin-2 mice and crossed it with the TDP-43 mouse that I said before that we generated in 2011. So both mutation create doubly transgenic mice. And I will refer to these mice as doubly transgenic for the rest of the presentation. So I will explain to you all the steps that we have to cross to do new mouse. So the first one is for sure to confirm the expression of your gene. So we introduce the Ubiquilin-2 gene. We have to be sure that the Ubiquilin-2 is in fact expressed in the cells. So for that, we can use Western blot. It's simply some gels that we put protein in, and we are able to use antibody to see the protein. You can see that our protein is expressed in the two transgenic mice, but it's not present in non-transgenic mice, so mice without the mutation. We also observed that our mice was expressing the protein to approximately 20% out of the level that the mouse has normally for Ubiquilin-2. So if we add both form of gene, the human one and the mouse one, it's a total of approximately 120% expression of the gene. It's interesting because we also observe that in human cases, the sporadic cases, we observe a 135% increase in the level of Ubiquilin-2. So our model is quite relevant to what we found in humans. So again, the second step was to analyze the TDP-43 pathology. So we have to be sure that we have a pathology, an inclusions pathology. So we took the spinal cord and the brain, and we are able to extract the protein from it and extract the protein from the cytosol and the nucleus. So like I said, the pathology is normally in the cytosol, so we observe an increase in the level of TDP-43 in the cytosol. You can see here that the bands are more intense than in the other groups. So we observe an increased TDP-43 in both spinal cord and brain, like we observed here, and this is significant results. So after that, we assured that TDP-43 was in fact localized into aggregates. So we did immunofluorescence, and we can see that this this co-localization of Ubiquilin-2 and TDP-43 into the neurons. So it showed that Ubiquilin-2, when we increase the level of it, it's creating a strong TDP-43 pathology as we see in human cases. So the third step was to analyze to be sure that we have a motor neuron loss and muscular atrophy also. So we use staining specific for motor neuron. We can see here spinal cord. It's maybe a bit thin for you, but the little dot are motor neuron. So if we count them, we see that there is less motor neuron here than in the other three groups. Also, we observed that the muscle fiber were reduced in size as if we compare it to the non-transgenic, and there is more white inside the picture. So this is conjunctive tissue. So it's when fiber are reducing, it's creating more conjunctive tissue. So we also observe a reduced size of the neurons and the nerve as compared to the non-transgenic. So we can conclude that we lose motor neuron, it's leading to muscular atrophy and nerve degeneration. So the first step was to analyze the phenotype of our mice. So we have to be sure that it's developing motor phenotype as we found in human. So we try different tests. The first one is the catwalk analysis. So it's basically a corridor. So we put the mice at one side of the corridor and it walked through the corridor. We can measure the strength left and also the base of support of the mice. So we observed that at 12 months and 18 months, there was a decrease in the base of support and in the strength left of the mice. So they have difficulty walking and extending their legs. We also use the Retiro test. This test is a classical test that we use in the ALS. We basically put the mice in a bar and we measured the time it takes before it fell down because the bar is running faster and faster. So we observed no big difference in our group except between 33 and 45 weeks of age. We observe small difference between the control group in green, in black, sorry, and the non-transgenic in green. We think that we didn't observe a big difference because the non-transgenic are really bigger and fatter because they do not have a muscular atrophy, so they fell faster because they are too much weighted. So it can change our data a bit. We also try another test, which was the pole test. So it's simply to put the mice on the pole, and we measure the time the mice take to descend the pole. If they have memory impairment, they take more time, or they completely fall from the pole. In our case, we observe an increased time to descend the pole in the doubly transgenic mice, but they did not fall from the pole, so they are not paralyzed. Finally, to analyze the memory symptoms from our mice, we use the passive avoidance. This is a test based on the natural tendency of the mice to enter the dark environments. So it's 2 chambers, 1 light and 1 dark, with a door between both. On the first day, we put the mice in the light room and it goes to the dark room. The second day, we do the same, but it receives a shock, a small shock in the dark room. In the third day, we put the mice in the light room and we measure the time it takes before going to the dark room. If the mice has a memory impairment, she does go to the dark room, sorry, because she does not remember receiving a shock. So at 7 months, we observed that our doubly transgenic mice has some memory impairment because they were going fast in the dark room. So they did not remember receiving a shock. And as compared to control, we were waiting almost 5 minutes before going to the dark room. The step 5 was, in fact, to analyze the inflammatory component of our models, because we know that there is activation of glial cells. So we observe, in fact, activation of glial cells in our doubly transgenic mice. Finally, the last step was to try to explain the mechanism underlying that. So like I explained before, Ubiquilin-2 normally make the link between the protein here in black, who has to be degraded. The protein are linked to ubiquitin protein before being degraded. So ubiquilin-2 bring the protein to the proteasome. This is a normal situation. When we increase the level of ubiquilin-2, I do not show all the results here because it would be too long, but I explain it simpler. So when we increase the level of ubiquilin-2, we saw that all the ubiquitin protein were in fact in the aggregates with TDP-43 and ubiquilin-2. So we think that the ubiquitin protein cannot be used by the proteasome to degrade the protein. However, when we increase the pool of ubiquitin, it's like the aggregates were saturated by ubiquitin. So the other ubiquitin can be used by the proteasome to degrade the protein. So this is an interesting mechanism who can possibly be used to target and treat the disease, because we just have to increase the pool of ubiquitin to favorize the degradation of TDP-43. So in first conclusion of our model, we can see that our model have a lot of features resembling the disease, as motor deficit, motor neuron loss, muscular atrophy, TDP-43 pathology, and age-related disease, and some cognitive deficits. So we think that our mice could be used to develop future therapeutic approaches. So when we have target a mechanism, when we have explained a mechanism and we have a model for it, we can try different drugs or kind of treatment. One of them to reduce the level of ubiquilin-2 could be all these mechanisms. We can use ASO or shRNA to reduce the level of ubiquilin-2. Or again, our antibody. In our lab, we have developed some antibodies against SOD1. And these antibodies are targeting SOD1, and they reduce its aggregation, its blocking, its toxicity. So it's like that we are coming to some kind of treatment to target some gene or some toxic protein. Sorry. Another way could be to have molecules to inhibit some pathway. In our lab, we have identified that the NF-kappa B pathway is toxic for the cells. It's an inflammatory pathway. And Jean-Pierre and the Imstar company in Vancouver are now developing a plant, a ashwagandha plant, with afferent A molecules inside. It's quite interesting in animal models, and we are now developing a clinical trial with that plant to test to treat ALS. So I want to thank Jean-Pierre and my co-director Nicolas Dupré for their supervision, and also the CIHR for my doctoral scholarship. So thank you to all, and if you have questions, please ask them.
Hello, Vincent. Sorry, I happen to be live at the Ottawa airport. Actually, I have a question for you. In all the work that you've put into reviewing the different mouse models, do you think that looking at them, there are a lot of different diseases that are genetic, that are funneling towards a sort of common endpoint, or do you think there might be an opportunity? Like, what's your opinion? I know everybody has a different opinion. Or do you think there might be some common mechanisms for treatment that could work for a larger group of people with ALS?
You are meaning genetic who are also implicated in other disease?
Well, so I'm thinking if you look at maybe the 10 or 9 different mouse models that now exist for ALS, and some of them with a lot of different ones, like, what are your thoughts on whether or not there might be a way that they all kind of funnel into a similar mechanism, or whether or not they probably should be individually treated? Or
I think that for sure all the mechanisms are quite related. If we take, by example, for sure TDP and Ubiquilin, but also FOS. FOS is also implicated in almost the same pathway as TDP-43. Probably it's a controversy subject, but the SOD1 into aggregates of TDP. I think that all the pathway are quite related, and if we want to treat the disease, I think we need more than one pathway treatment. We didn't try any clinical trials yet, or even animal treatment who we target more than one pathway. And I think it can be the key. I think my work is showing that at least a few mechanisms are linking together. So.
Great, thank you. And I guess only one other question I have is, so what's the next step for you? You're looking at therapeutic mechanisms to potentially use your model. And then if you are, what other model do you think you would use next, or want to look at next to sort of confirm what you might be seeing in your TDP-43 Ubiquilin-2 model?
Yeah, yes, in fact, we are probably beginning one other project. I don't want to give too much detail on it, but we want to probably to target the expression of ubiquilin or the impact of ubiquilin on ubiquitin protein. So we want to try like genetic modifications. I don't want to say the precise one, but to target ubiquilin. And we will try for sure in our doubly transgenic mice, and we want to try maybe an SOD1 case, because I want maybe to show that ubiquilin is also implicated in SOD1 case. It's not proven yet. And also we will maybe begin a collaboration with François Gros-Louis, who have a model of skin who came from patients, so it can be a great sporadic models. So with all that, we can target almost all cases with different mutation or sporadic. So we want yeah, we want to target ubiquilin-2 overexpression as therapeutic avenue.
Great. Well, thank you very much, and really appreciate your time, and thank you everyone for joining us today. If you have other questions, you can send them to us, and we'll forward them on to Dr. Picher-Martel to see some of those answers. But otherwise, if anyone's sticking around for the French webinar, we're going to log off and log back on for that. And appreciate everyone, and have a great rest of your day.
Thank you, and I have put my email in the presentation if you want to send me some questions.
Thanks very much.
Thank you.
Thanks. Okay, bye-bye, everyone.